You have selected the experiment. Experimental animals routinely used in pharmacological research. Objectives: to understand the specific characteristics and uses of experimental animals in pharmacological research. Click Next to continue. Introduction. Preclinical phase of experimental pharmacology depends upon the use of experimental animals. Even though the guinea pig has become synonymous with experimental animals, many other species are equally useful in the study of pharmacology of drugs or new drug formulations. Experimental animals can be of different species like frog, rat, mouse, rabbit, guinea pig, hamster. Frog. Biological name Rana tigrina. Specific characteristics: frog is a cold-blooded amphibian. It has 3 chambers in its heart, 2 auricles and 1 ventricle. Uses in experimental pharmacology. Various tissues isolated from frog like rectus abdominis muscle, heart, gastrocnemius, muscle sciatic nerve preparation, etc. are used in experimental pharmacology. Different drugs acting on central nervous system, neuromuscular junction, and heart can be studied using frog and its isolated tissues. Whole frog is also used in screening of certain drugs like local anesthetics. Use of frogs in experimental demonstrations is banned. Common strains used: Rana esculenta, Rana pipiens, and Rana temporaria. Rat. Biological name Rattus norvegicus. Specific characteristics: rat is a warm-blooded rodent. It can't vomit and does not possess the vomiting center. It has no tonsils and gallbladder in its body. Hence, it can't be used in screening of the drugs having activities on vomiting center or gallbladder. Rat is an omnivorous animal. It shows resistance to the effects of cardiac glycosides. Uses in experimental pharmacology. The isolated tissues like ileum, trachea, heart, anococcygeus muscle, stomach strip, vas deferens, uterus, and phrenic nerve diaphragm are used in experimental pharmacology. The rat and its isolated tissues are used in the screening of drugs acting on central nervous system, cardiovascular system, estrous cycle, mating behavior, gastric ulcers, and acid secretions in stomach. It is also useful in the hormonal assays, teratogenicity, and carcinogenicity study, hepatotoxicity studies, and acute and chronic toxicity studies. Rats can be trained to carry out certain functions like lever pressing in response to a reward or punishment. Hence, they can be used in screening of psychoactive drugs. Common strains used: Lewis, Sprague-Dawley, Wistar, and Porton. Mouse. Biological name Mus musculus. Specific characteristics: mouse is the most widely used animal in different toxicity studies. It is a warm-blooded rodent. Mice are very sensitive to the sedative effects of hexobarbital. Uses in experimental pharmacology. Isolated tissues from the mice are rarely used in the experimental pharmacology because they are very tender. Still, in certain cases, vas deferens and ileum from mice can be used as isolated tissues for experimentation. Mice are used in acute and subacute toxicity studies, cancer and genetics research, immunity and transplantation-related experiments. They were earlier used in the bioassay of insulin. Common strains used: Swiss albino, bulb C, and C57BL6. Rabbit. Biological name Oryctolagus cuniculus. Specific characteristics: rabbit is a warm-blooded mammalian. Some strains of rabbits are resistant to the effects of atropine because they have higher concentrations of atropinase enzyme in their blood. They do not show Dale's vasomotor reversal because they do not possess the adrenergic vasodilatory nerve. In this species, coitus itself induces secretion of luteinizing hormone in females, which leads to ovulation. Another hormone, progesterone, is known to block such ovulation. Uses in experimental pharmacology. Isolated tissues like duodenum, ileum and heart are used in experimental pharmacology. Rabbits are mainly used in the pyrogen testing, in bioassays of antidiabetics, neuromuscular junction blockers and sex hormones. They are also useful in the study of teratogenicity of drugs. Bioassay of adrenaline can be performed using rabbit ileum. Strains used: New Zealand white, Himalayan black. Guinea pig. Biological name: Cavia procellus. Specific characteristics: guinea pig is a warm-blooded rodent. Its name itself has become synonymous with an experimental animal. Uses in experimental pharmacology. It is mainly used in the bioassay of histamine. The isolated tissues like ileum, tracheal chain, vas deferens and heart are used in experimental pharmacology. It is also useful in screening of drugs for the treatment of tuberculosis, cholera and amoebiasis. They are used in the study of drugs like spasmogens, spasmolytics, local anesthetics, bronchodilators, and digitalis. The drugs inducing anaphylaxis and those affecting the hearing activity are also screened using guinea pigs. Strains used: Duncan Hartley. Golden hamster. Biological name: Mesocracetus auratus. Specific characteristics and uses in experimental pharmacology. Golden hamsters are nocturnal mammals and live in deep burrows in their natural habitat and are considered to have their origin in Syria. Their food includes grains, fruits and insects. An adult hamster weighs about 150 grams and is 15 to 20 centimeters in length. Hamsters are susceptible to chemically and biologically induced tumors and are preferably used in testing anti-cancer activity of drugs. They have a larger surface area in the cheeks, and this forms pocket-like structure. These cheek pouches are used for implantation of carcinogens. They are also used in immune system-related studies. The organ rejection rate in this species is lesser than other rodents. Apart from this, hamsters are used in behavioral testing, toxicity testing. Strains used: Syrian hamster, Chinese hamster, European hamster, South African hamster.